1. Paddy, Oryza sativa, 2 N equals 24. Rice is the staple food of more than 60% world population and most of the people of SE. Asia. About 90% of rice grown in the world is produced and consumed in Asia. It is a crop of wet tropical climate and also grown in humid regions of subtropics. In India, rice occupies the first position among the cereals in respect of both area and production. Rice contains less protein 6 to 7% in white rice and 7.9% in brown rice and contains 2 to 2.5% fat which is lost during milling. Hulling percent in rice is 66% or 2/3 of paddy. In general, cereal crops required about 400 to 500 liters of water for the production of 1 kilogram of plant dry matter. But rice consumed 10 times more water, i.e. 5,000 liters of water to produce 1 kilogram rice grain. In rice field, to prevent the nitrification and leaching losses, N fertilizer should be incorporated in reduced zone so that the process of nitrification is delayed. Rice field submergence in 5 cm deep water during the reproductive and grain formation stages is beneficial. Rice grain relatively rich in lysine, which is 4% of total protein fraction. The main protein in rice is oryzanin. Propanil, stam F34, is the most widely used post-emergence herbicide in rice. Area and distribution. Area. India greater than China greater than Indonesia. Production. China greater than India greater than Indonesia. Productivity. USA greater than Japan greater than China. In India. Area. West Bengal greater than UP greater than Bihar. Production. West Bengal greater than UP greater than Andhra Pradesh. Productivity. Punjab 34 Q per hectare. Classification. Genus Oryza have 24 species out of which 2 are cultivated. 1. Oryza sativa, diploid, 2 N equals 24, it has 3 main geographic races. A. Indica, grown throughout tropics and subtropics in India and characterized by tall stature, weak stem, elongate grains, droopy leaves, low productivity and irresponsive to high input conditions. B. Japonica, grown in temperate and subtropical regions of Japan and characterized by short stature, sturdy stem, narrow erect and dark green leaves and responsive to intensive input condition, having highest productivity, grain shape are oval to round in shape. C. Javanica, Balu rice, grown in Indonesia, few tillers, wild form of rice, has many awns and long duration crop period. 2. Oryza glaberrima, it is grown in West Africa only. Origin. Indo-Burma region, SE. Asia. Rice is self-pollinated crop, fruit type is Chiopsis. The grain or Chiopsis is tightly enclosed by the lemma and polea, collectively called the hull. Test weight, WT, of 1000 grains, is equals 25 grams. Rice stem is commonly called the haulm or culm, which made up of a series of nodes and internodes. Inflorescence of rice is panicle, which is a group of spikelet. Climate, C3, short day plant and required hot and humid climate. Soil, clay and clay loam are best suited for rice cultivation. Acidic soils are good for rice cultivation. Most suitable pH range is between 4 to 6. Average temperature for plant growth is 21 to 37 degrees Celsius. In India, mostly rice cultivated on low land condition. Rice is semi-aquatic plant grown best under submerged condition. Under these conditions, the atmosphere oxygen is transported by erenchymatous tissues of leaves. Seasons. Oz. Sowing time April-May. Aman, Kharif, sowing time June-July. Boro, Spring, sowing time November-December. In northwest part of country only Aman, Kharif, crop is taken due to falling of temperature in winter. Varieties. First dwarf variety of rice developed in world is TN1 in Taiwan in 1964-65. First high-yielding dwarf variety developed in world is IR8, by cross between Peta D. G. Wu General. It was the first known photo-insensitive variety to be released by International Rice Research Institute in 1966. Literally meaning of D. G. Wu genus, brown-tipped short-legged. First introduced variety of rice in India was Taichung Native 1, TN1, in 1964. IR8 was the second introduced variety in India from the IRRI, Manila, Philippines, in 1965. First Indian high-yielding earliest semi-dwarf variety Jaya released in 1968, evolved by Dr. Shastri, and it's a cross between TN1X type 141. It's good yielder as compared to TN1 and IR8 both. Hence called miracle rice in India. Cuttack, Odisha, evolved two varieties, Malinga and Masuri. Mutant dwarf varieties of rice, Jagannath and Sattari. Varieties suitable for direct seeding, Bala, Kaveri, Nagina 22, Pusa 2-21. BLB, bacterial leaf blight tolerant, TKM-6. Salinity tolerant, IR8, Lunashri. Suitable for waterlogged area, Jalmagan. First hybrid of Basmati in world, PRH10. First hybrid of rice in India is CORH1, 1994. Variety released from IARI, Noran 8, 18, Pusa 2-21 and Basmati 370, Pankaj, IR20. Pusa Basmati 1, its world's first high-yielding dwarf variety under quality rice has been developed by IARI through convergent breeding. Other varieties, Padma, for upland, Pankaj, waterlogged, Ratna, Chambal, Sita, Sabarmati etc. Seed rate. Broadcasting, 100 kg per hectare. Drilling, 60 kg per hectare. Hybrid rice, 15 kg per hectare. Depog method, 1, 5 to 3 kg per square meter. R times P distance, 20 by 10 centimeters. Fertilizer management. NPK ratio 100, 60, 60 and 25 kg zinc sulfate, at last puddling, is generally recommended in rice for high-yielding varieties. The waterlogged puddle soil develops two zones. The upper zone, 1 to 10 mm, a thin surface layer which contains oxygen is called oxidized zone, and immediately below the oxidized layer lies a reduced layer which contains no oxygen is called reduced zone. Use of nitrate fertilizer in paddy field should be avoided due to more susceptibility to loss of nitrogen through leaching and denitrification. Hence, in lowland the deep placement of the ammoniacal form of nitrogenous fertilizer in the reduced zone has been proved to be efficient method of nitrogen application than other methods. If we have to use nitrate fertilizer in lowland then we should be incorporated in oxidized zone only. Nitrogen use efficiency in rice, 30 to 40%. Biological nitrogen fixation. It has been estimated that the total nitrogen requirement of rice could be brought down by about 25 to 30% through biofertilizer, algae. In addition to blue-green algae, BGA, the association of the water fern azolla with the blue-green algae Anabaena azole also helps in biological nitrogen fixation. Azotobacter, a non-symbiotic nitrogen-fixing bacteria also fixes atmospheric nitrogen biologically in upland rice field. Kharia disease. Zinc deficiency causes kharia disease in rice usually at nursery stage. The tip of the zinc-deficient leaves first become yellow and then dry and plant remain stunted. Zinc deficiency is marked by the appearance of rust-colored or brownish-red coloration on the surface of the outer leaves. First reported by Y.L. Nene in 1966 at Pantnagar, Uttarakhand. It is managed by spray of at 5 kg zinc sulfate at nursery stage. Or foliar spray of 0.5% zinc sulfate in early stage of crop. Browning of rice. Iron toxicity in rice is known as browning of rice. Iron toxicity is the major problem to rice production in waterlogged acid sulfate soils of altisols and oxisols soil orders. The beginning of iron toxicity is characterized by the appearance of purple coloration at the leaf tips and finally has a scorched appearance. Falguna variety is tolerance to Fe toxicity. Silicon in rice. Silicon is regarded as essential nutrient in rice production. Silicon deficient plants are soft and droopy. Silicon supply mostly required during the panicle initiation stage because during this stage root activity is relatively reduced and the rate of transpiration is high. It has been noticed that silicon provides a thick cuticle silica layer which functions as a barrier against attack by insect pests. Silicon deficiency can be corrected by application of basic slag at 1,5 to 2.0 T per hectare. Because basic slag is the most common source of calcium silicate. Weed management. Echinochola crushgoli, barnyard grass, and E. Kernum, wild rice, are major weeds in rice. The weed problem is less in lowland transplanted rice compared to upland direct seeded. Herbicides used in rice crop. 1. Bentiocarb, satin, at 2 kg a.I. per hectare is applied as pre-emergence of weeds. 2. Propanil, Stam F34, at 3 kg a.I. per hectare is applied at 1 week after transplanting or post-emergence of weeds. 3. Butachlo, Machete, at 2 to 3.0 kg per hectare is used as pre-emergence, i.e. weeds have not emerged in field. It is effective to control both annual grasses and broad-leaved as pre-emergence of weeds because emerged weeds are not killed. 4. Anilefos. It is a pre-emergence and early post-emergence selective herbicide used for control of annual grasses, sedges and some broad-leaved weeds in transplanted and direct-seeded rice. Bushening. It involves cross-plowing the young crop, 4 to 6 weeks after sowing with a light country plow in 5 to 10 cm standing water, once or twice. Purpose behind this to control weeds, optimize crop stand and provide soil aeration. Puddling. It is a process of destruction of soil aggregates by mechanical force in soils with excessive moisture content. Puddling destroys the soil structure which hampers root penetration and the development of the succeeding upland crops grown in rotation with rice. Puddling increases the bulk density of soil about from 1.4 to 1.7 grams per cc. It is done with equipment, known as puddler. Objective of puddling. To reduce percolation losses of water. Main object. To reduce weeds infestation. To increase nutrients availability. Critical stages for water. Telling stage. It comes 2 to 20 days after transplanting. Booting or late panicle initiation stage. Swelling of the flag leaf sheath is called booting. It is most critical stage for water in rice crop. Depog method of rice cultivation. This method was introduced from Philippines. In this method, seedling becomes ready for transplanting on 12th day after seeding. It comprises rising of a dense stand of seedlings without contact of soil. Rate for nursery sprouted seeds at 3 kg per square meter on wooden planks, trays, concrete floor, and seedbed covered with polythene sheet. Hence, 25 to 30 m squared nursery area is sufficient for 1 hectare. Hybrid rice. In the last decade of 1970, hybrid rice was firstly developed by China by using cytoplasm male sterility fertility restore gene system. After China, India is the second country to commercialize hybrid rice of APRH1, APRH2, AP, KRH, Karnataka, and MGR1, TN. PHB 71 is the only hybrid rice which is released by private organization. Golden rice. Term coined by Ingo Potricus. Beta-carotene-rich gene inserted in rice through genetic engineering. That fortified the vitamin A content in rice. It is transgenic or GM rice. Shri, system of rice intensification. This methodology was developed by Henry de Laulani in Madagascar in 1980s. It is an agroecological approach for increasing the productivity of irrigated rice by changing the management of plants, soil, water and nutrients and cultivating of rice with as much organic manure as possible. Basic principles in Shri are significantly reducing plant population, irrigation quantities and improving soil conditions by using less or nil herbicides or chemicals. Benefits include 20 to 100% or more increased yields, up to 90% reduction in required seeds and up to 50% water saving. With Shri, average yields of rice are 6 to 7 tons per hectare, compared to conventional method 2-3 tons per hectare. Shri mainly adopted for irrigated upland or rain-fed rice as well as other crops such as wheat, sugarcane, millets, mustard, etc. Shri is more feasible for the farmers who have small landholdings and need to get the highest yields possible from their available lands. In Shri, wide spacing planting, 25 by 25 centimeters are adopted and transplanting of young seedlings at 2-leaf stage, 8-12 days old. Nursery area is 100 square meters enough with seed rate of 2 kilograms acre or 5 kilograms per hectare. Demerit of Shri is higher labor costs in the initial years for individual seedling transplanting and weeding. Processing of rice. 1. Milling. The aileron layer and embryo are removed during the polishing of rice grain. There is a loss of vitamin B, particularly thiamine, the deficiency of which causes the disease called beriberi in those persons who continuously eat polished rice. 2. Polishing of rice. The removal of very fine bran clinging to milled rice and smoothing of the surface is called polishing. 3. Parboiling. It is a hydrothermal process in which the crystalline form of starch is changed into an amorphous form on account of irreversible swelling and fusion of starch. Important questions. Sowing time of Oz, autumn rice, April-May. Sowing time of Boro rice, November-December. Rice inflorescence is known as panicle. Rice is a type of plant, short day plant. Cardinal temperature of rice is 30 to 32 degree. Hulling percent of rice is 65 to 66%. Fruit of rice is known as chiopsis. Widely used nitrogenous fertilizer in rice, ammonium sulfate. Optimum pH of soil required for growing rice is 4 to 6 pH. Mat type nursery is related to crop, paddy. Test weight of basmati rice, 21 grams. Most critical stage of water for rice, booting stage. Biofertilizer for rice is azolla. Seedling ready for transplanting in depog method after sowing. Days 11 to 14. Most dominant weed species in rice, Echinochola sp. White eye of rice due to Fe deficiency. The gas emits from rice field is methane. Akiochi disease in rice due to H2S toxicity. Highest nitrogen losses in the rice crop by denitrification. Para and utera cropping system is closely related to which crop? Rice. Parboiling of rice conserve the vitamin B12. Seed rate in depog method for rice, 3 to 4 kg per square meter. Area required for seedling preparation in depog method, 25 to 30 m squared. Miracle rice of India is Jaya. Gene responsible for dwarfness in rice, DGVU gene. Spacing for rice sowing of seedling, 20 by 10 centimeters. Kharia disease of rice is due to zinc deficiency. Reclamation disease of cereals due to copper deficiency. A rice grain is said to be extra long if it is 7 mm or more.